Hi, I'm James Verdeer, and welcome to the American Institute of Biological Sciences Bioscience Talks, which is a forum for integrating the life sciences. Today's episode is the next in our In Their Own Words oral history series, in which we talk with scientists who have made great contributions to their fields, particularly in the biological sciences. This week's guest is Dr. John Burris, who is Emeritus President of the Burroughs Wellcome Fund. He's also a past president of AIBS. Dr. Burris, thank you very much for joining me today.
It's my pleasure. I look forward to chatting.
I look forward to it as well. And our first prompt is, when did you first know that you wanted to work in the life sciences?
Just as all things start when one is young, but just by way of background, my father was a professor of biochemistry at the University of Wisconsin, and my mother had a master's in nutrition. And so the setting, the environment in which I grew up was a very strongly science-oriented environment. So that was sort of a background at all times, or from day one, so to speak. I think it was really crystallized by 2 summers that I spent, one after my sophomore year in high school and the other after my junior year in high school. At that time, the National Science Foundation had these programs called Science for high school students. I'm sure there's a more formal name. But what they were, and they were a wonderful experience. First one was at Cape Hayes Marine Lab in Sarasota, Florida. And that was, you have to realize I grew up in Madison, Wisconsin, and so the opportunity to spend a summer studying marine life in Florida was an amazing one. And the programs were super good programs. The one at Cape Hayes involved, there were 11 of us. We lived together in a motel, and then every day we took classes and then did research at Cape Hayes. And Cape Hayes was a small marine lab, and its specialty was sharks, pretty exotic for a sophomore in high school from Wisconsin. And the leader of the lab was a very famous woman, Eugenie Clark, who had written a book, Lady with a Spear, and she had worked on shark research. And so we not only learned all about the marine life and went to classes, but we went out on the boat and, believe it or not, caught sharks and then brought the sharks back if they were, tried to keep them alive for behavioral studies, if not, dissected them. So spending 8 weeks studying marine life was pretty strong reinforcement for wanting to be a scientist. You have to realize that this is the era of Jacques Cousteau. That would have probably been the summer of 1964 that I did that, and this is when everybody was excited about Cousteau and the oceans. And then the following summer, I went to another NSF program at Scripps Institution of Oceanography in La Jolla. And that was very interesting because Scripps was a part of UCSD. UCSD was basically in its infancy, and we lived in a college dormitory. That program was a little larger, 20 plus kids, but we took classes and then were assigned to do research. And I was assigned to, or chose to do research with a plankton expert, John McGowan, who was professor at Scripps, and then one of his graduates, 2 of his graduate students, a fellow named Charlie Miller and Pete Wiebe. And so not only did we sit on the beach every day, but I went on a 2-week cruise to Baja California. So it's sort of hard when you're exposed at an age like that to 2 experiences like that, not to say, I want to be a scientist. So I'd always wanted to be a scientist, and this reinforced it. I went to college and majored in biology, did the sort of normal lab stuff. Summer after my junior year in college, I spent the summer at a marine lab in Finland and studied that one. I worked as an independent investigator. I think they weren't quite sure what to make of this American college student that came over, and so I got a lab and studied vertical migration and zooplankton in Finland. And so the hook had been set, so it was pretty certain I just loved science and was intrigued by it, and was one of these fortunate people that were put in an environment that just reinforced it. And so it was pretty clear. And that's not to say I couldn't have deviated from the path, but it was pretty certain, and I just enjoyed it. I spent a year at the University of Wisconsin after graduating in an MD PhD program. Decided I really wanted a PhD, not an MD, so I went to graduate school at Scripps Institution of Oceanography in La Jolla, and that's where I got my PhD. So by the time you're getting a PhD, you're pretty certain you want to be a scientist, but mine is not a very twisted path. It's a pretty straight path to science.
So I'm curious, what sparked that brief urge to pursue an MD? What were you thinking about at that time?
Well, this was at the height of the Vietnam War and student protest. I graduated from college in 1971. We'd had 1.5 year where they'd actually, at the end of the year, sent all the students home, and there was an enormous amount of protest. And there was a lot of feeling, and I was part of it, that you wanted to be relevant, you wanted to do good, and I mean that in a positive way. And so medicine seemed to be a natural place for me to go, but I was in an MD PhD program, so I always was interested in doing research with a medical bent, and figured that an MD would be helpful. I still think to this day, to some extent, of course, an MD does give you some advantages in terms of research on humans and sort of being in the forefront of understanding what diseases are relevant and also human suffering in the sense of that. So that it was an MD PhD program, and I didn't do much of the MD part of it. I actually at Wisconsin took a bunch of limnology courses and realized that really my interest was much more in that end of things. Wisconsin had a very strong limnology department at that time with John Magnuson and Art Hassler. So it was sort of something that intrigued me, and then that offered the opportunity to go on to Scripps. But I think that was a sort of the social environment of 1970, 71, 72. I mean, people that were alive then will certainly remember that if they were college students.
That makes sense to me. So after your graduate studies, what came next in your career? How did you get that first job?
I was very lucky. I did not have to do a postdoc, and that was nice. I got a job at Penn State. They advertised for a botanist, and I didn't know much about plants, but I knew a lot about photosynthesis because my research as a graduate student was on photorespiration in marine algae, and primarily studied the zooxanthellae, the unicellular dinoflagellates that live inside corals, and looked at the symbiosis between the two of them. And I had a cruise, a couple cruises, but one was to the Great Barrier Reef, in which I spent 8 weeks anchored on an island off the reef and looked at coral symbiosis. And so that gave me a reasonable feel for plants because I was looking at photosynthesis and photorespiration. So I got a job at Penn State as a plant ecologist, and I guess that was a rather broad definition of what I'd done, but I ended up teaching introductory ecology and introductory botany, so I think I learned hopefully as much as the students, or at least a little bit more. But I was just very lucky. I published some papers and had a reasonable success as a graduate student, and I was able to get a job right away as an assistant professor. So moved to State College, Pennsylvania, right as I finished my PhD in 1976.
Okay, and if you don't mind, I'd like to take you back for just a moment to that, that Great Barrier Reef. That was 8 weeks anchored just off the reef or on the reef?
Well, there are a series of islands. They're called continental islands, just 30 some miles, I guess, offshore, with reefs around them. Great Barrier Reef is huge and stretches forever. And this was an island called Lizard Island, and it was really a neat place. At that time, nobody was there. There was a hermit lived on the island, but that was it. It's now I haven't been back, but it's now a luxury resort. But when we were there, we had this island and a mountain that supposedly Cook had climbed to help chart his way through the passages of the reef, and it was an Alpha Helix cruise. The Alpha Helix was a National Science Foundation ship that was operated by Scripps, but the expeditions were not necessarily Scripps-based expeditions. They were scientists supplied to NSF, and a professor, a fellow named Ed Tolbert at Michigan State, had set up this cruise, and so I was fortunate to be on that cruise as a graduate student, and my major professor was also on the cruise. And interestingly enough, my father was on the cruise. My dad was the chief scientist for the expedition, had been a graduate student of my father's. So my dad went and he studied nitrogen fixation on the reef. Not much of that had been done before, and he had developed, along with some of his postdocs and visiting scientists, something called the acetylene reduction method. And that's a way to look at acetylene reduction as a measure of nitrogen fixation. It didn't require N15 isotope, which is the way that had been the only other way really to do it. And he'd done that forever. He had built a mass spec and himself in the 1940s, and had studied with Harold Urey, who was the Nobel Prize winner at Columbia, and who discovered the heavy isotope of water. And so my dad had gone to Columbia, postdoc learning about N15, came back to Wisconsin as professor and built a mass spec and measured N15. So he was the guru of nitrogen fixation. I mean, he was a member of the National Academy of Sciences and National Medal of Science winner, et cetera. But it was fun to have him on the cruise. It was sort of out of his environment, the marine environment, but it was nice to have him along on the cruise also.
Yeah, that does sound enjoyable. And I'm wondering, is there a lot of cross-pollination of ideas, among the various researchers who work on quite clearly a diversity of different topics, in those kinds of settings?
Absolutely. I mean, the cruise was fun because there were a lot of, as I said, different people, terrestrial scientists, one from Georgia and from Michigan State and all over. And I ended up collaborating with a scientist, Clanton Black, who was at the University of Georgia, who I probably would not have ever had any cross-pollination or very limited interactions with. But you're on there, somebody's doing a fun project or something interesting, and you may be able to bring isotope knowledge, or you may be able to provide some thoughts on the enzymes. And the Alpha Helix was set up very specifically for what you have alluded to, which was an area for a group of scientists to physically work together. And we had almost daily seminars in which people would talk about what their research was, just informal talks, so you'd know what A, B, or C was doing. And then you sort of take it from there. You say, oh, wow, that's interesting. That's kind of like what I'm seeing. And then we would go out together in the field. And there was a scientist from UCLA, Len Muscatine, who was really sort of the expert coral biologist. And he sort of would help everyone learn more about the natural environment, and then all these biochemists, because for the most part they were biochemists, and on the trip would bring the ideas and say, well, maybe if we do this, this, or this. So everyone who has been on a Helix cruise, and you'll cross paths with them, I suspect, a little bit. There was a very famous scientist, Per Scholander, who was sort of the father of the Alpha Helix in terms of setting it up. We'll all say it was one of the most productive times that they ever had, because they were on this ship. And in our case, the ship was anchored off this island, and so it was really literally a floating laboratory without even the distractions of being at sea and steaming and all that sort of stuff, which takes a lot of time when you're an oceanographer. You go from A to B to C. None of that was the case. We all were on the cruise, and it was both Americans and Australians, which was very nice. So that was good to have Australian scientists involved, because again, that's a group one would have had very little contact with.
Yeah, and I think that's an interesting thing, and it kind of feeds into one of our other questions, which is how have things changed over the course of your career. But that certainly sounds like one, in terms of the ability to do collaboration. I'm guessing that talking to Australian scientists in the 1970s would be something that would have to be conducted over a telephone and a shaky international line at best, whereas now it's probably quite different.
Yeah, absolutely. And also the physical contact. I mean, we go diving and collect a specimen for someone to use in an experiment. I mean, you really, it's like a cauldron of ideas and organisms and all sort of mixed together, and you're not going anywhere. I mean, maybe people say, well, but for 24/7, you're together. I mean, we could go. You could take a skiff on shore if you wanted. You could hike around a little bit on the island, or go. But basically, there you are. So it was good, and it was just a good opportunity to interact.
It does sound so. So why don't you tell me a little bit about your time at Penn State? What was that era like, and what sorts of things were you working on?
So Penn State was and is a very large university, and it's kind of interesting because Scripps didn't have any undergraduates. They were all graduate students. There were undergraduates on the upper campus at UCSD, but you didn't have to do any teaching. You got paid as an RA, research assistant. I made an effort to TA one term in an introductory botany course up on the upper campus, so I had a little experience. But you sort of land at Penn State, and I still remember my first class. There were 396 students, and there were 396 students because that was the largest lecture room at Penn State. So I sort of was plopped down in front of them. And so I learned a lot of botany and a lot of ecology very quickly. And then, so it was a sort of standard teaching research situation where you had to teach introductory courses. And with time, I was able to teach the more advanced classes, but I started out teaching the big introductory ecology and introductory botany class, and then built up a lab. I mean, it was one of those, you got a little bit of money to start up, pretty standard in terms of how research still works, except whereas today scientists in the biomedical fields at least start with $1 million startup. I think mine was probably more like $40,000. But I set up a lab, and then I started to recruit graduate students. And then, as now, you are told up front you're going to be judged on publications and grants, and your teaching is something you have to do, and you have to do well. But you're not going to get tenure from it. You have published in grants. So I sort of knew the system, and so you just dive in. And I was very lucky to get some really good undergraduates initially. Penn State has a very diverse population of kids from the state of Pennsylvania. It's sort of the default institution that Pennsylvania's young people go to, and so some really terrific undergraduates, and then with time worked to build up more graduate students. But it was a pretty standard career path for anyone, which was publish, get funded, and I got a nice grant, a couple of nice grants from NSF, and that was really important because I could support graduate students. And with one of the grants, I went back to the Great Barrier Reef as the chief scientist, not on the Alpha Helix, on a boat that we chartered from New Zealand. And spent 8 weeks, again, not just on lizard, but a series of spots on the reef. And that one was fun because I was able to put the crew of scientists together myself, and I thought it was was a very productive time, and we had most of an issue of a journal, and so that was very helpful in my career. So
that sounds incredibly rewarding. I'm wondering then, what feeds into the decision to, step away from that, kind of traditional academic role, and how do you find yourself at the National Research Council?
Sure. That was sort of an interesting story. I enjoyed teaching, I enjoyed the research, but I also enjoyed policy and interacting with people, et cetera. So I went to a garden party. The Academy each year has what's called a garden party, where the members come and they bring guests. And so I drove up from State College with my wife and up to DC and met my parents there at the garden party. And I was talking to someone and telling him what I was doing. And he said, well, they're starting a new board at the National Research Council called the Board on Basic Biology. And that might be something that would work for you. We sort of talked. So I was intrigued and I applied. And so my year after I got tenure, I went to DC in this job with the thought that if I liked it, I would stay. If I didn't like it, I would go back to State College. But I loved it. And it was really what intrigued me. Fascinating. We're working right away in the first year on a variety of things, a big study of a whole field of biology, a committee chaired by Peter Raven, who I'm sure you know. And almost overnight, I got to meet everybody in the field, from whole organism people like Peter and Tom Eisner to Francisco Ayala, a long list. And this new board flourished. A very nice fellow, Art Kellman, who was a plant pathologist at Wisconsin, was the chair of the board. And I loved it. So I severed the golden handcuffs, made a decision that I was going to leave the world of research and go into policy. And the Academy at that time was a great place to do it. And I got a lot of support and mostly just fascinating, interesting people. I mean, just great stuff. And I had a wonderful board with lots of support from them. And a whole bunch of fun studies came to the board. Our board put on a joint symposium with the Smithsonian called the Biodiversity, and that actually is where the word was coined, was the title of that. One of the staff officers, Walt Rosen, came up with it, and Ed Wilson compiled the book called Biodiversity. And it was to see a word go from biological diversity to biodiversity and become such a part of the language. And worked a lot with Peter. And as you well know, because I'm sure you've interviewed Peter, I think you did, in fact, he's an enthusiastic, interesting fellow. Then along the line, I ended up being the study director of a study looking at mapping and sequencing the human genome. And Bruce Alberts chaired it. And on that committee was Jim Watson and Dan Nathans, and for a while, Wally Gilbert and Sidney Brenner. And those were just the Nobel Prize winners. It was and we made a recommendation that the government should map and sequence the human genome. And 2 weeks later, NIH started the Human Genome Initiative. So it's probably the one time in one's life where one makes a recommendation, someone picks up the recommendation, and now, let's see, that was 85, 6, somewhere in there. 35 years later, they're sequencing every virus that comes down the line. But you have to realize that back when we did the study, there was a lot of opposition to it. It was felt that it was just repetitive work and not that important, and there were lots of skeptics about it. And ultimately, they think nothing of sequencing a genome, but keep in mind, people got Nobel Prizes for four base pair sequences versus, what we do now. So it just repeated things of that sort. I did a study on use of laboratory animals, and then I moved up to head of the Commission on Life Sciences. And in that portfolio was all the follow-up on the atomic bombs in Hiroshima and Nagasaki, the Radiation Effects Research Foundation, and we ran the U.S. end of that. In fact, the Academy, Truman had put the Academy in charge of the Atomic Bomb Casualty Commission back in the when he became president in the 40s. And so that involved learning all about radiation and going to Japan, because part of the Radiation Effects Research Foundation funding came from the U.S. and the other half from the Japanese. So there are a whole host of wonderful things, and that was much too long an answer to a short question. So maybe I'll let you go next.
No, not at all. I mean, that's, it sounds like very exciting stuff and a real diversity of topics. Is that one of the major appeals of this type of work, is that you're able to kind of move to one topic and then explore it in depth, but then move to something completely different, more or less?
Absolutely. If you track what I've done in my career, you'll see that I just retired from the fund where I worked for 11 years, but every job before that had been 8 years long. And each one provided a set of just intriguing things and different. I've always liked working with people. I've enjoyed all the opportunities. And it's not that I get bored, it's just that I've come to find things that are very different and very different life experiences. And so I've fortunately had a family that's tolerated a number of moves and willingness to live in different places, and seems to have survived fine from it. So I think when opportunities come, as I said earlier about tenure being golden handcuffs, I think if you have a chance and something looks interesting, you should see what you see as the merits and demerits of it, but be willing to try new things, because certainly from my perspective, all these different things have been both rewarding and fun.
Okay, and I remember it was shortly after this era that you were the president of AIBS, but at the same time you were doing that, you were at Woods Hole, correct?
That's right. I went to the Marine Biological Lab in 1992. So I had been at Penn State, and then I went to the Academy. Yeah, I went to Woods Hole in 92. My life seemed to be tied to institutions. The MBL is and was a great independent organization, and that was a terrific 8 years. It was a magnet for some of the best courses each summer, great summer research institution people. We went from a couple hundred people in the winter to 1,000 people in the summer. Great courses, great science. But the model and the problem was that it was wonderful and everything, but we didn't have any endowment to speak of. And so we kind of lived a hand-to-mouth existence. We collected some rent and collected some money from the courses. And so we aggressively worked to raise money. We started a capital campaign, which ultimately reached its goal. And we built on the amazing loyalty of the scientists in the summer who'd come for $1 million every year for 20 years. We built on the strength of the winter program, particularly Ecosystem Center, but other good year-round scientists like Shinya Inouye and others. And then we raised money, and we worked very hard, and we came out of it in good shape. At the end of the time, we had excellent board, really strong board chair, Shelley Siegel, And it just was an exciting and great time.
How did you find that, sort of fundraising experience? Because, obviously we're moving very far away from, certainly research, and then also, kind of even away from other sorts of administrative tasks in some senses.
Yeah, fundraising, first you have to have a good person who does the legwork for you, and I had a very good director of development at the MBL. So I like to talk to people. I enjoy it. It didn't bother me to ask people for money, because I think what I was asking for was worthy of support. We traveled a lot of it with my wife, Sally, who was very, very good at this and very personable individual. We traveled all over, primarily the East Coast, because that's where the money is for the MBL, New York, Washington, Florida in the winter. And we made presentations and talked to people and got to know people. And we would get to know people even better in the summer when they were there for 3 months. So quite frankly, I didn't dislike it.
Absolutely. And what drove your next move? What was the impetus for moving along from the MBL?
I had we had very much enjoyed Woods Hole, and we really liked the MBL, but I guess it was an impetus to try something different. My eldest daughter had gone to Carleton College in Northfield, Minnesota, a very fine, small liberal arts college, and she was enamored of the experience, and I became intrigued by what a great job small liberal arts colleges do in training, educating, whatever term, young people to be independent, particularly many of them in science. The hands-on science is taught at those places. I just thought it was a great model education. So I thought, well, what do I want to try? I want to try something. I'd raised the money at the MBL, and it was time to try something else. And so, as I said, it was primarily probably the model of my daughter's experience at Carleton that said, well, let's look at small liberal arts colleges. So that's how I sort of then morphed into being a college president at Beloit College.
And what was that early experience like? What are the first few days on the job? When you take on that type of role.
Well, I think most people, I hope most people, when they take on a job there, is they get to know with whom they're going to work. And so a lot of time working with the faculty, with the students, with the board. Small colleges are very personal. And I think for some kids, for some students, they're great, and for other students, they're not. But if you want an individual experience where you get to know your professors and things of that sort, that's a great place to go. And that's really, when you go to a job like that, it all goes back to getting to know the people with whom you're going to work, and that's the students, the faculty, the alums. So there are a lot more hands-on, personal sort of work, certainly when you start. As time progresses, of course, it becomes more rigid in the sense that you've got to do X amount of fundraising, and you've got to do Y of this and Z of that. But when you start, you're just sort of an open book, walking around, learning, meeting people, talking to people, get an idea of what they think is good and what they think is bad, what should be changed, what shouldn't be changed, and all those sort of things.
What was probably the biggest challenge, if you had to name one, from that period?
Oh, the biggest challenge is, quite frankly, you never have enough funding, never have enough money. You're constantly working to balance the costs with what you can charge. If you look at the balance sheets of almost all small colleges, they're tuition driven, but the revenue from tuition is not enough to cover the cost, your fixed costs or your salaries, which are probably 80, I don't know, a while, but 80+% of the operating budget is some sort of a salary cost, maybe a little less than that. And so that means that you're sort of running in place. You're always trying to find donations. You don't get grant money, so you have to raise money. You're also looking at what's essential and not essential. There are a few of the small schools that have big endowments, and so that's less of an issue for them. But for 90% of the small schools, and I can't conceive of how hard it must be during COVID times, you're just working as hard as you possibly can to be able to both maintain the institution and also change it, in the sense that if you're a static institution and nothing is changing, you're going to be left behind, and they're going to start a spiral down.
So it's a case of, kind of making those difficult decisions where that some constituency is going to be frustrated with the decision, but it's kind of something you just have to do in order to keep the organization economically afloat.
Yep. I think and I mean, AIBS, a good example of that, right?
Absolutely. You're in a state of constant change, and if you're not constantly changing, you will most certainly be left behind.
Yeah.
So, what was the cause for your next leap? This is obviously to the Burroughs Wellcome Fund, and where not having an endowment is not an issue. How did that experience differ, and what sorts of things were you doing there?
Sure. I had served as chair of one of the Burroughs Wellcome Fund advisory committees, the one that looked at funding in North Carolina for pre-college science, technology, engineering, math, STEM education. And I'd done that for a number of years, and then my term had ended, but I knew about the fund. And I really thought it was doing wonderful work, because it had this mission of supporting biomedical research and supporting K through 12 STEM education just in North Carolina. It was a nice balance, a nice organization, a nice place. So when the opportunity arose, I was ready to stop raising money. Even though I said I don't mind raising money, I was ready to stop it. I went to the fund, and a small organization with a good-sized endowment, and able to give away money in a very peer-reviewed fashion. I love the way, every money wasn't what I wanted, where I wanted to give it. There were programs. We had very stringent requirements, and we did peer review. So we'd get 100 applicants, and we'd give out 12 awards. And what the fund did and does is they would actually bring the applicants to North Carolina and interview them. So they'd bring in twice as many applicants as awards that they were ultimately going to make. So I just liked the process. My predecessor had done a really good job in establishing this peer review system, and the fund had gotten, because the company was sold, the fund got this money from the Wellcome Trust, who owned the company, and the Wellcome Trust provided a $400 million endowment, and so we didn't have to raise money. Now, the Wellcome Trust, you may not know, is a British trust. It's the second largest charity in the world after Gates. They probably have 30 billion or something. So they gave us the 400 million, and we spun off as a totally independent foundation. Previous to that, had been part of the company, and the company was sold. Glaxo bought the company, and then so it was total independence. It was a board of directors who were all scientists. It was this wonderful organization in the sense of independence. There was nobody, as we jokingly said, named the Burroughs or Wellcome on the board. So it was really a very peer-reviewed, careful organization with hardworking staff and a board of scientists with no particular axe to grind. And we had enough money to give away $35 to $40 million a year, which was not huge, but enough to, I think, make a difference.
Most certainly. What sorts of projects and organizations were you funding during your time there?
We were funding individuals within areas. So we funded people that worked in infectious diseases, and we've done that for many years. And of course, that's timely with COVID. But we funded individual scientists. Almost all were young, either postdocs or assistant professors in infectious disease. We funded MDs who were doing research, so physician scientists. We had a program for physician scientists. We had a program for quantitative scientists who were doing biomedical research. So by quantitative, that's not quantitative physical scientists, engineers. They had to have a degree in something other than biology, and they were applying it to biomedical research. We funded parasitology. So we chose areas, and then we funded young people, junior scientists within those areas. And we included also in the, several other programs. That was the basis. And then the other big program was pre-college science. And there we funded museums and schools in out-of-school learning. So we funded a variety of things in North Carolina in education. And then we had I think a very nice long-term program in diversity where we funded postdocs from underrepresented minorities. So it was a nice mix from kindergarten to advanced postdocs, professors, etc.
Okay, and I was hoping we could talk a little bit about a thread that I've sort of noticed you've developed in our discussion today and has recurred throughout your career, which is the importance of STEM education. And obviously for those who are going to pursue careers in STEM and become research scientists, et cetera, but also there seems to be an emphasis in many of the things you've done on ensuring that the STEM experience is good and valuable for those who are not necessarily going to wind up in one of those careers. And I'm just kind of wondering if that's something that has been a deliberate effort or, if it's just something that's kind of happened along the way.
Well, they're all absolutely deliberately. I've long felt that you have sort of to assume everyone should, and this sounds arrogant, but assume that everyone should know science or STEM. Understanding that from the beginning, most of those people will not be scientists, will not be mathematicians, will not be engineers. But the fact that they don't pursue a career is not a failure. The fact that they have some comprehension of science, and by that I mean STEM, is an asset for society. I've always thought that the most important thing is that we not say, well, the general public is ignorant. What we say instead is it's our responsibility to provide an education in which understanding of science, and by that I don't mean to run experiments, et cetera, but have comprehension of what science, how it operates, what the scientific method is, some quantitative field, orders of magnitude, things of that sort, so that every U.S. congressman has some feel for science, as well as providing programs like I was exposed to in high school, which were pretty sophisticated science and were 8 weeks and all that sort of stuff. I want all your children and my grandchildren and my children to have some feel and understanding of science so that they can not understand all the details about what messenger RNA is, but have some feeling of what is happening, for example, in this vaccine and how it's working and how it's protecting us, just as an example, or have some feel for climate change and what human, again, not having to know all the details, but just having some feeling of what anthropogenic means and what the sources are and how climate change is being driven. And that's my Pollyannish goal, but I think that's so important.
No, I think that's an excellent answer to the question. And now our last one. Do you have any advice for young scientists who are entering the field right now? And we can expand that out and make it into a general STEM question as well. What sorts of things should they be thinking about and focusing on?
Well, I think there are two things. Number one, be prepared constantly for change. If you think just of computers and the that you would have no idea, even when I started my career 50 years ago or whatever, where there were no computers, or if there were computers when I was in college. But I mean, the day-to-day use of computers, the fact that I'm holding a supercomputer in my hand right now as I talk to you on the telephone. So that's change. That there's constantly going to be change, and you have to be prepared for change and adaptation to change, and understand that change is coming, and change is constant. And then the other thing, though, is, and I found this has stood me in good stead, if you understand the fundamentals, and this is most pronounced whether it's in biology or chemistry, you understand sort of the fundamentals, that gives you a base in which you can go back to it. If you understand like the fundamentals of chemistry, you can go back to it. Or if you understand some of the fundamentals of biology that haven't changed, and defining fundamentals, evolution would be a fundamental in biology, but it's also change. But if you can educate yourself so you understand the basics, many of the basics, that will stand you in good stead in your career. So be prepared for change, but understand some of the core tenets of the sciences and the scientific method, so that you always, when you seem you're swamped by change or whatever it is, you can always go back to those core concepts and figure out what may be happening or what is happening.
I think that sounds like great advice that we would all be well served by heeding. Dr. Burris, thank you very much for joining me today.
James, thanks a million.
And that concludes this episode of Bioscience Talks. Just a reminder, the journal Bioscience is published by Oxford University Press on behalf of the American Institute of Biological Sciences and is made possible by the support of our members and donors. Thank you, and talk to you next time.